Good morning, students. Today we are going to discuss about chromosomal inheritance. So here we see in the next slide, what is chromosomal inheritance and what is the role of inheritance in chromosome. So chromosomal theory of inheritance was described by Suttan and Boveri in 1902. In earlier case, we know that up to Mendelian law of inheritance, we found the law of dominance, law of segregation, and law of independent assortment. But after that, the factor is named as gene, and chromosome plays an important role where linkage group of traits are found that express in link form as reported by Suttan and Boveri in 1902. But how many linkage group in the human species are found? That is another question. In 1920, Morgan et al., that means another scientist also involved in chromosomal theory of inheritance, that genes are arranged in a linear sequence on the chromosome. They can be mapped. So chromosomal theory of inheritance, if you described in detail, it was proposed by Walter Sutton, full name is Walter Sutton, and Theodore Boveri. And they work out the chromosomal movement during meiosis. That we know that during meiosis, the chromosomes are distributed in the gametes in haploid form, and chromosome movement is actually worked by Sutton and Boveri. The movement behavior of chromosomes was parallel to the behavior of gene. The chromosomes movement is used to explain Mendel's law. Before that, Mendel could not explain or relate with the factor. That means chromosomes are involved in gene movement that cannot be explained earlier, but Sutton and Boveri proved with the help of chromosome movement the law of Mendel. The knowledge of chromosomal segregation with Mendel principles is called chromosomal theory of inheritance. So what is chromosomal theory of inheritance? How the chromosomes segregate or move, that is explained with the help of Mendelian principles is called chromosomal theory of inheritance. According to this theory, chromosomes and genes are present in pair in diploid cell. As you know that the somatic cell of an organism always in diploid, so chromosomes and genes are present in always in pair. Homologous chromosomes separate during gamete formation, that is meiosis. As you know, in germ cell, meiosis occurs in sexually reproducing organism. So the two chromosomes separates in homologous pair during gamete formation, and fertilization restore the chromosome number to diploid condition. As you know, the male and female gametes are haploid, and after fertilization they are converted to diploid form. After that, this chromosomal theory of inheritance also described by Thomas Hahn Morgan and his colleague conducted an experimental verification of chromosomal theory of inheritance, and Morgan used in his experiment the fruit flies. It is the common name, and the scientific name of fruit fly is Drosophila melanogester. And here you see the Drosophila melanogester with male and female type of fruit flies. So why Morgan selected Drosophila or fruit fly? He selected Drosophila because it is suitable for genetic studies. As we know that Mendel only performed hybridization experiment with the help of pea plant, but in case of Morgan, he used the insect fruit fly, and it is suitable for genetic studies. Secondly, it is grown on simple synthetic medium in the laboratory. That means it can be grown in artificial medium easily. They complete their life cycle in about 2 weeks. Within 2 weeks, they completed their life cycle, and a single mating could produce a large number of progeny flies. So in the production of progenies, and the study of the character can be made easy. Clear differentiation of male and female flies. Male and female flies are quite different in their characters, and many type of hereditary variation can be seen with low power microscope. So these are the advantages why Morgan selected fruit fly or Drosophila in his experiment. So chromosomal theory of inheritance can be explained with the help of meiosis. We know that meiosis completed in two phases, meiosis 1 and meiosis 2. In meiosis 1 anaphase, here you see during anaphase, the chromosomes separate, homologous pair chromosomes separate. One chromosome from paternal and one chromosome from maternal come together, and in anaphase they separate. So in meiosis 2, the chromatids separate. So in this way, each chromosome move to the different germ cells to express their characters. This is one probabilities. Here, one long orange color chromosome with short green color chromosome and long yellow color chromosome with short red color chromosome. Another probabilities, one long orange color and short red color chromosome, other yellow color with green color chromosome. So in both cases, in meiosis 1 anaphase and meiosis 2 anaphase, they segregate. First, the homologous pair separate, and second, the chromatids separate. So during gamete formation, each possess or bear only one chromosome with distinct characters. So this is also the same case. Actually, chromosomes, when they duplicates or separates, it actually starts from cell cycle. As we know that G1 and G2 phases are the phases where the cell component increases along with the cell organelles. But in S1 phases, the duplication of the chromatin material occurs. And these are called bivalent. That means in meiosis 1 or prophase 1, we found that leptotin, zygotin, pectitin. In the leptotin stage, they become paired up. One from maternal chromosome, other from paternal chromosome paired up. There is a synaptical force between them, and after crossing over, they separate in the anaphase. Anaphase, so first you see the two gametes from progeny produced from these cells, two gametes or two cells, one chromosome move here, another chromosome move here. And in the second metaphase or meiosis 1, and the two chromatids separate, and the other half they synthesize from the information of this DNA or chromosome. Next, chromosomal inheritance, the characteristic we actually, the Morgan actually took, one the white eye of the Drosophila, and the that is male characteristic, and the red eye Drosophila, it is the female characteristic. So X denotes, X are XX when paired together. Here X denote wild type red eye, and X also denote allele white eye. So homozygous means 2 X chromosome, and hemizygous means 1 X chromosome with another 0. That means here, as you know, in case of human, 1 X chromosome. In case of male, 1 X chromosome and another Y chromosome is there. But in case of insect, we denote this one is as 0. So here you see the wild type contain red eye. Here it contains 2 X chromosome. And in case of male Drosophila, 1 X chromosome and another Y chromosome. That means Y does not carry any heritable characters. So here it produces two kind of sperm, one with this chromosome, other with this chromosome. And the female produces two type of egg, one this egg, another this egg. So when this egg paired with this sperm, then it produces red and white together. Here also red, here also red and white, and here also red. So here all daughters are red heterozygous. That means although the white and one chromosome bear white eye, another chromosome bear gene for red eye, but the red one is dominant. So in case of female, they are heterozygous. Chromosomally they are different, but they expresses the dominant gene, that is red eye. But in case of male, here it is also red eye. They exhibited the characteristic of red eye because in X chromosome the red eye gene move not in the Y chromosome. So it is the two daughter or the male offsprings are always with red eye. But here we call it is hemizygous because only one X chromosome bear the characteristics. Thank you. This is the end of our class today. So we discussed detail about our chromosomal theory of inheritance. because earlier we found that in Mendel's law only he studied about the plant, pea plant, that is Pisum sativum, but it cannot be explained theoretically how chromosome related with the inheritance of gene, and it was discovered by the Suttan and Boveri that the factor denoted by the Mendel can be denoted as gene in 1902. And after that, Morgan experimentally proved the chromosomal theory of inheritance and relate it with the Mendelian principles by using Drosophila melanogester. So I think this is the end of our slide today. And if there is any doubt, you can ask me in our next class. Thank you, student.